Hello, myself Santosh Kumar from Jodhpur. I love my passion of the teaching. Why? There's a big reason behind it. We have looked around ourselves, we have seen many type of irregularities, we have seen the problems, we have seen the corruptions in each sectors of our daily life activities. So how can we remove the corruptions, the problems, the irregularities, all the sectors that is behind us? That can be possible only just by educating the nation, not by imposing new laws and rules and regulations to over the public. We can change the luck of any country, we can change the map of any country by educating the nation. So the education has a power that can change the all the things that is created, that can solve the problem, that can give a new way, a new look, a new basically the futures to the nation. So I have loved to teach the nations, I have loved to teach, to give the knowledge, to literate the people, that hence they will not repeat the problems, hence they will not create the problems. So I love my passions of teaching. Today we want to discuss about the genes regulation. The topic is related to the 12th grade biology, chapter 6, molecular basis of inheritance. So how today we want to discuss about the regulation of gene expressions, that how the gene is expressed. We have a many cellular activities inside our cells that is responsible for the performing any type of functions. Now the question arises that how the function is regulated. We have learned from the basic standards that our all the functioning is connected toward the DNA, and the DNA is responsible for the controlling any type of functionings, either that is metabolically or either it is physiologically functioning. Now we want to describe it how it is actually regulated inside our cells. Generally, what did you mean by the gene? Gene doesn't mean the DNA. Gene doesn't mean the DNA. Am I correct or not? Yes, I am correct. Because why? The gene, there is not any connection of the DNA and the gene. We cannot say that DNA is a gene. We can say that the particular part of a DNA is represented as a gene. How? Because the part of a DNA that is responsible for the formation of the mRNA, further it will form the protein or enzyme that is responsible for creating or that will responsible for performing any type of functioning. In that way, that portion of the DNA is known as by the gene, because the DNA contains that much part that is not responsible for performing any type of activity. Hence we cannot say that all the portion of the DNA is known as by the gene. Further, the portion of the DNA that is responsible for the performing any type of activity inside the cell, it is known as by the gene. Further, the genes are the two types. One is constitutive genes and another one is non-constitutive genes. The constitutive genes are known as by the housekeeping genes. These are expressed continuously inside the cells. Because what is their final product that is responsible in every time inside the organism's body. Hence, they activate all the time inside an organisms. These all the regulations are occur at the cellular level. Let me explain you what did you mean by the gene expression. The gene expression simply means switching off or expression of the certain genes by inhibiting the expression of another genes. That whole mechanism are known as by the gene expression. Suppose that there are the two genes. One gene is responsible for the formation or for the metabolism of a certain product. Another gene that inhibits it to metabolize or to perform its functions. If that gene is inhibited or that gene is suppressed in such a way that the next gene will express itself, that whole mechanism is known as by the gene expressions. Now as you know that the ATP, it is responsible for the any activity that is occurring inside our cells. We can generally call it ATP is a currency of our cells that is responsible for the creating any type of functioning. Further, the gene of glycolysis, the gene of glycolysis is responsible for the breakdown of the glucose. These both genes are required continuously inside our body, hence these are continuously formed at the cellular levels. Also, in that way, the genes which is responsible for the formation of ATPase or glycolysis are continuously forming, hence they are known as by the constitutive genes. Another category of the gene is non-constitutive genes. Those genes, they are not expressed continuously inside the cells or at a cellular level. They are expressed only when, if they are required. If they are not required, they will not be expressed, are known as by the non-constitutive genes. Further, the non-constitutive genes is for the two types, inducible genes and another is a repressible genes. What did you mean by the inducible genes? these type of the genes are generally remain in the repressed conditions or switch off condition at the cellular level. They will become functioning or they will become switch on if the product that is metabolized, if it is present inside the cell. In that condition, that they will perform their functions, otherwise they will not perform their functions. The product that is metabolized, it acts as an inducible molecule to start that type of a gene, hence that type of expression is known as by the inducible gene. Further, another one that we are calling it is a repressible genes. These genes are continuously expressed inside the organism's body at cellular level. When these genes are formed much than their required amount, in such a way these genes are goes outside of the cells. In that way they produce a chemical interaction that is responsible for the switching of the expression of their genes. That type of a gene expression is known as by the repressible genes. So let me clear you one another things. The inducible genes are remain in switch off condition. It will perform when the product that should be metabolized, if it is present. These are expressed themselves till the product is formed much or more quantity than the required one. In that condition, they will replace their expressions. So these are the two types of genes. It is a category of non-constitutive genes. Now the gene expression is depend upon the certain factors like metabolism, physiological and environmental factors. These are the factors which are responsible for the regulation of the gene. If there is any interference between these factors, then the expression of the gene regulation can be inhibited or that they can be altered. Further, the gene expression inside the cells in eukaryotes can be occur at the various level. At the transcription level, what did you mean by the transcription? The process of the formation of mRNA from DNA. The process of formation of mRNA from DNA. Sorry, yes, I am right. It is known as by the transcriptional level. Further, the processing level. What did you mean by the processing level? When the mRNA is formed, when the RNA is formed from the DNA, it is not known as by the mRNA because it contains a functioning portion along with a non-functioning portion, generally known as by the introns and exons. So that RNA is known as by the heterogeneous RNA. That in that way the RNA, the portions of the RNA that contains the non-functional part should be removed. That process is known as by the processing level. So the gene regulation could be occur at the processing level. When the mRNA is formed, it will be transferred from nucleus to the cytoplasm because the formation of the protein occurs inside the cytoplasm. So at the transport level or at the transportation level, the gene expression can be occurred. And last, the translation level. When the mRNA reached inside the cytoplasm, with the help of ribosomes, it is responsible for the formation of the protein. So in that way, on that point, the gene expression could be occurred in the eukaryotes. So these are the 4 points, these are the 4 paths at where the eukaryote can express their genes. Now let me explain you the operon concept. What did you mean by the operon? Operon simply means the gene, the portion of the DNA which is a self-regulating. That means it can switch off or it can remain switched on conditions without depending upon the another genes, or the portion of the DNA which is responsible to express to basically we can say that to regulate any type of activity without the help of any other portion of the DNA. So that portion of the DNA is known as by the operon. The operon concept was given by the Jacob and Monod. They and the operon genes in the, if we want to explain the beta galactosidates are the metabolism, how the metabolisms of the lactose occur inside the eukaryotic cells. In that conditions, that portion of the DNA that is responsible for the metabolism of lactose. So in that conditions, the operon consists of the 4 type of gene. One is regulatory, another is promoter, third one is operator, and last one is the structural gene. Let me explain you the functioning of the each gene. What is the regulatory gene? It generally forms the protein known as by the repressor that will combine or that will basically bind with the operator gene, and it inhibit the translation procedure. In the case if the lactose is present inside the cell in such a way that the lactose combined with the final product of the regulatory gene, generally known as by the repressor, and it inhibits its binding with the operator gene. In such condition, operator will remain switch on condition, and the RNA polymerase enzymes will bind on the promoter. In So the promoter, after it all, it will move and on, move ahead on in such a way the structural gene should be it should be transcription, and the translation process will be occur at the structural gene. In such a condition, Z gene will be responsible for the formation of the beta galactosidase enzymes, which is responsible for the metabolism of lactose into galactose in and glucose. And Y gene is responsible for the formation of the permease enzyme, which is responsible for to increase the permeability of the cell membrane to intake the lactose. Where the A gene releases the trans-acetylase, which is responsible for the transmission of the acetyl group from the cobalt acetyl to the beta galactosidase. So that is a concept of the operon. So it is known as by the inducible gene expression, because if the product is present, then it will express. If the product that is metabolized, if it is not present in such a condition, it will not express itself. So it is all about the gene expression. If you liked it, if you want to know more about it, then comment me. I will make another video to for you. Goodbye. It's Santosh Kumar.